Bats have been on Earth for over 50 million years. They survived ice ages and mass extinctions. So why are bat viruses suddenly showing up in human outbreaks more than ever? In this video, we will show you the reasons bat viruses spill into humans and why one virus called Nipah can be so dangerous. From SARS to COVID-19 to a virus killing up to 75% of those it infects. Nipah. And in 2025 alone, fatal cases emerged in Bangladesh and India, more than past 2 decades combined. And now, scientists are racing against time to stop what could be the next pandemic. Bats are uniquely suited to host viruses. In 2024, scientists identified 24 new Nipah species in Australian flying foxes alone, revealing 3 entirely distinct viral clades. Most have never been isolated or studied, but they're all closely related to Nipah and Hendra viruses. Unlike most mammals, bats can fly, an activity that pushes their metabolism to extreme levels, with heart rates up to 1,000 beats per minute during flight. Over millions of years, this forced bats to evolve hyper-efficient immune systems that tolerate viruses without destroying them. Recent genomic studies in 2025 show bat genomes are significantly enriched with immunity-related genes, specifically for viral detection, antiviral responses, and inflammation regulation. Instead of destroying viruses, bats coexist with them through dampened inflammatory responses. This makes bats natural viral reservoirs for dozens of deadly pathogens, often completely asymptomatic carriers, capable of hosting multiple viruses simultaneously. Viruses that barely affect bats become catastrophic when they spill over into humans. Pandemics aren't becoming more frequent because bats are invading our habitats, we're invading theirs, and forcing fundamental changes in their behavior. A landmark 25-year study published in 2022 in Nature revealed something alarming. Bats are no longer just temporarily responding to habitat loss, they're permanently changing their behavior. Climate change and deforestation have destroyed bat-feeding habitats, forcing fruit bat colonies to relocate permanently into agricultural areas near human populations. What used to be temporary stress responses, like leaving forests during food shortages, have become permanent behavioral patterns. Key drivers of spillover: deforestation destroying winter-flowering trees that prevent spillover events, industrial farming bringing livestock directly into bat territory, urban expansion creating persistent bat-human overlap, wildlife trade and wet markets creating viral mixing zones, climate disruption causing nutritional stress that increases viral shedding. The study found that when winter-flowering remnant forests provide adequate food, spillovers can be prevented. But as climate change disrupts these seasonal patterns, the windows of protection are shrinking. Each disruption increases the probability of zoonotic spillover, and recent research shows viral shedding peaks during bat breeding seasons, when juvenile bats with waning maternal antibodies become highly susceptible. Both SARS and COVID-19 trace their origins to bat coronaviruses, and new 2025 research reveals how they reached humans. A groundbreaking study published in Cell in May 2025 used genetic analysis to track sarbecovirus evolution. The findings were startling. The bat viruses that became SARS-CoV-1 in 2002 and SARS-CoV-2 in 2019 both traveled over 1,000 kilometers from their origin points to reach Wuhan and other urban centers, far faster than bats could naturally migrate. The dispersal pattern is consistent with one thing: the wildlife trade. Horseshoe bats typically forage within just 2 to 3 kilometers and disperse slowly. But both pandemic viruses left their origin areas less than 10 years before emerging in humans, a speed only possible through human transportation networks. What made COVID-19 uniquely devastating wasn't just deadliness, it was efficiency. Highly contagious before symptoms appear, airborne transmission in enclosed spaces, global travel networks enabling instant worldwide spread. But there's a bat-borne virus that terrifies epidemiologists even more. Meet Nipah virus. It kills up to 75% of those it infects. First identified in Malaysia in 1998, Nipah has caused near-annual outbreaks in Bangladesh since 2001. India has seen recurring spillovers in Kerala state since 2018. The 2024 to 2025 outbreak wave. In 2024, India reported multiple Nipah cases with a shocking 100% fatality rate in some clusters. Bangladesh documented 4 fatal cases through August 2025, bringing the total to 347 confirmed cases since surveillance began, with an overall case fatality rate of 71.7%. January 2026 brought 2 new confirmed cases in West Bengal, India. Both healthcare workers infected while treating patients, highlighting the virus's dangerous ability to spread in hospital settings. How Nipah spreads. Primary route: bat saliva or urine contaminating date palm sap and fruits. Amplification: infected pigs and other domestic animals spreading to humans, human to human. Through respiratory droplets, saliva, and bodily fluids. Nosocomial transmission: direct infection of healthcare workers. Unlike many viruses, Nipah spreads efficiently inside hospitals, turning outbreak response into a high-risk operation. Recent research shows the virus can contaminate hospital surfaces, creating multiple transmission pathways. When Nipah doesn't kill, it devastates. Initial symptoms: 4 to 21 days after exposure. High fever, severe headache, muscle pain, vomiting, sore throat, dizziness, rapid progression to acute encephalitis, brain inflammation, acute respiratory distress syndrome, seizures leading to coma. There is still no FDA or EMA approved cure for Nipah virus as of early 2026. Current treatment reality: intensive supportive care is the only option. Neurological monitoring in isolation units. Experimental antivirals tested only during outbreaks. Survival depends entirely on quality of intensive care. Long-term effects for survivors. The horror doesn't end with recovery many Nipah survivors face. Permanent neurological damage and cognitive impairment. Persistent personality changes. Severe memory loss. Recurring seizures requiring lifelong treatment. Delayed reactivation. The virus can remain dormant and reactivate months or even years later, causing relapse encephalitis. Recent long-term studies of survivors in Bangladesh reveal that neurological complications persist, in most cases, fundamentally altering quality of life permanently. So if Nipah is this deadly, why hasn't it caused a pandemic? Because, so far, it lacks one critical element: efficient, sustained human-to-human transmission. Current Nipah outbreaks show 2 distinct patterns. Acute respiratory distress syndrome predominant outbreaks, 2001, 2007, 2018, 2023, show human-to-human transmission, higher spread. Encephalitis predominant outbreaks, 2019, 2021, 2024, no secondary transmission observed. This suggests the virus exists as multiple strains with varying transmission capabilities. However, viruses evolve. Every spillover event is another opportunity for Nipah to acquire mutations that enable efficient airborne spread. Research shows fruit bats harbor not just one Nipah strain, but multiple circulating variants. And we've only isolated and studied a handful. The 2024 Nipah R&D Roadmap update emphasized the critical window is closing. Climate change is increasing spillover frequency, and we're discovering the virus in wider geographic ranges. But there's hope, and it's accelerating faster than ever. December 2025, breakthrough. The University of Oxford launched the world's first Phase 2 Nipah vaccine trial in Bangladesh, the epicenter of annual outbreaks, the Chadox-1 Nipah B vaccine, using the same platform that saved 6 million lives during COVID. It is being tested on 306 participants. The vaccine received prime designation from the European Medicines Agency in June 2025, fast-tracking development. October 2025. The Serum Institute of India partnered with Oxford and CEPI to manufacture the world's largest investigational reserve, up to 100,000 vaccine doses that could be deployed during future outbreaks. July 2025. A second vaccine candidate, PHV-02, received $17.3 million in CEPI funding to advance to Phase 2 trials in Bangladesh. This live attenuated vaccine offers protection after a single injection. The challenge? Nipah outbreaks are too small and sporadic for traditional Phase 3 efficacy trials. Scientists are pursuing alternative regulatory pathways using surrogate immune markers and animal studies, similar to how Ebola vaccines were approved. Multiple vaccine platforms are now in human trials: viral vectors, mRNA, subunit vaccines, all racing toward a 2030 target for licensure. Preventing the next pandemic doesn't start in hospitals, it starts in ecosystems. What's working right now? In Bangladesh, the UK Health Security Agency is implementing school-based education programs, teaching children about Nipah transmission. When kids understand not to drink uncovered date palm sap, they educate entire families. Protective measures proven effective. Covering date palm sap collection sites with bamboo skirts prevents 90% of bat contamination. Protecting winter flowering forest remnants that provide bat food during critical periods. Reducing livestock bat overlap through strategic farm placement. Strict hospital infection control with contact, droplet, and airborne precautions. Enhanced surveillance during peak risk seasons, December to May. Rapid outbreak response teams deployed within 24 hours. The bigger picture. Climate change is compressing the timeline. As we lose more bat habitat and disrupt seasonal flowering patterns, spillover windows are expanding. Bats aren't the enemy. They pollinate crops worth billions, control insect populations, and sustain entire ecosystems. Over half of North American bat species face severe population decline in the next 15 years from white nose syndrome and climate change. The real danger is a world where ecological balance collapses faster than science can respond. In January 2026, West Bengal saw 2 healthcare workers contract Nipah while saving lives. In Bangladesh, 4 people died in 2025 from drinking contaminated date palm sap. In Kerala, outbreaks now happen almost like clockwork. The pattern is clear. As long as we destroy habitats faster than we build defenses, spillover events will accelerate. But for the first time in history, we have vaccines in advanced trials, global surveillance networks, and proven prevention strategies. If bats have carried these viruses for millions of years, and pandemics are only happening now, what does that say about the choice we face? Do you think the Nipah vaccine will arrive in time? What would you do to prevent the next spillover? Share your thoughts in the comments and subscribe for science-backed topics.